immunoelectrophoresis. Remember, we are talking about soluble antigen only. Immunoelectrophoresis, immuno समझ गए होंगे, एंटीजन एंटीबॉडी, इलेक्ट्रोफोरेसिस यानी फोरेसिस मतलब मूवमेंट. दिस इज मूवमेंट अंडर द इन्फ्लुएंस ऑफ इलेक्ट्रिक current, right? So in the above case, what is happening, the antigen antibody are diffused, right? But in this case it will move under the influence of electric current. So what we do, we actually do this experiment. So same experiment, यहाँ पर सेम वैसे ही है, वेल्स है, एक में एंटीजन है, एक में एंटीबॉडी है, and we put it in an electrophoretic tank, right? In the electrophoretic tank where there is a suitable buffer is present and terminals that the cathode, the anode and the cathode terminals, right, minus and plus terminals होंगे, and there is supply of electric current. अब supply of electric current के लिए we have a power supply. अब आपका power supply में ये connect हो जाएगा simply. This electrophoretic unit is used for the plasmid this was DNA separation or nucleic acid separation. We same electrophoretic tank use किया जाता है. So this is connected, connect कर दिया गया है, right? And we supply the current and specific volt, say 100 volt हमने current voltage दे दिया and now 50 current है. So what will happen in this case? In this case, now you should know the charge on the antigen. In this case, the charge on the antigen should be confirmed because the antigen will move towards opposite. So if it has a negative charge, it will move towards positive, right? But antibody with a no net charge, it will move on every place, on every side. So this antigen will move fast and this line of precipitation will appear fast, so within 1 hour or 2, right? So this is called as immunoelectrophoresis. Specifically, this is called as counter current immunoelectrophoresis. Counter current immunoelectrophoresis. काउंटर करंट इसलिए बोला गया है क्योंकि यहाँ पर एंटीजन और एंटीबॉडी अपोजिट डायरेक्शन में मूव कर रहे हैं एंड द एंटीबॉडी मूव्स अपोजिट द फ्लो ऑफ करंट एंड गिव अ रिएक्शन. दिस इज अ काउंटर करंट immunoelectrophoresis. And in general, immunoelectrophoresis is performed when you want to, if you know the charge on the antigen and you get that result earlier as compared to this diffusion. So immunodiffusion and immunoelectrophoresis are also some preliminary screening tests which are performed when the antigen is soluble. But if the antigen is particulate or cellular, then we know that we perform agglutination test. अब देखिए जैसे double immunodiffusion किया गया, but in case of agglutination, the nature of antigen is particulate. You can see here, these are the antibody, these are the antigens. अब आप देख रहे हैं किस तरीके से बाइंड होंगे and this forms a lattice structure, mesh like structure. It gives a visible reaction. Positive negative को यहाँ पर compare किया गया है and then it is showing that it may be positive, it may be direct agglutination, it may be indirect agglutination. ये further immunology में आप डिटेल डिस्कस करेंगे. Different types of agglutination, this we have discussed, hemagglutination, right? Hemagglutination, when RBCs are involved, this is hemagglutination. You can perform this agglutination test on a 96 microtiter plate also, and you can see the clumping or the button shape like structures. आप जब देखेंगे कि agglutinate होगा तो वो नीचे एक बटन जैसा स्ट्रक्चर दिखाई देगा. So you can perform it on ELISA plate as well. This is a passive. This is called indirect. Indirect का मतलब होता है when antigen is now directly binding to the antibody. Antigen is first coated on a this your latex. Yeah, this is important again. This is important. It can be asked a question. Now question can be, let me write it on the copy. Now it is clear जो मैंने अभी तक बताया कि if the antigen is soluble, if एंटीजन इज सॉल्युबल. अब क्वेश्चन ऐसा आता है कि virus sample is provided to you, the nature of the sample, a virus sample is soluble in nature, which test you will perform to identify. So you will say we'll do precipitation test. That's it. The question is, can we identify soluble antigen by agglutination test? Right? Can soluble antigen be identified by agglutination test? Because we know that in agglutination test the nature of antigen is cellular or particulate. How we can identify soluble antigen by agglutination test? So in this case what we do, we incubate or coat the soluble antigen over a inert material. अब हम सबसे पहले क्या करते हैं, एक inert material लेते हैं. This is an inert material with no charge, no by this, and it is latex bead. Now what we do, we coat the latex bead with the soluble antigen. This is like this way. We call it a coating of antigen over inert material, say latex beads. Now it becomes cellular. अब ये बन गया cellular या particulate. अब इस जो आप पीपीटी में देख रहे थे, ये देखिए, ध्यान से देखिए क्या किया गया है. इस केस में दिस इज inert material, latex bead, and these are the antigens. It is coated over the inert material. Now this antibody is binding to the antigen. Since the overall it become a particulate, you can see in a clumping form. So लिखा है measurement of antibodies to soluble measurement of antibodies to soluble antigens. So this is how you can make a soluble antigen particulate, and this is called as indirect agglutination. इसको कहा गया है indirect agglutination. Then ELISA we have discussed, this is not related to you. Yeah, fine. So now this happened, this will bind, and since it is more than one bead, से बहुत सारे beads होंगे तो ये ऐसे bind हो जाएगा. यहाँ पर भी एक और इस तरह से होगा, and it forms a agglutinated structures. Right? So ultimately what is happening, होता क्या है, देखिए यहाँ पर क्या है कि दो एनालाइट है, एक है एंटीजन और एक है एंटीबॉडी. ये जो एंटीजन और एंटीबॉडी की बाइंडिंग हो रही है, राइट, ये हो रही है वहां पर जहां पर इन दोनों का कंसंट्रेशन सेम है, जिसको हम कहते हैं जोन ऑफ इक्विवेलेंस. जो कॉन्सेप्ट है किसी भी सीरोलॉजिकल टेस्ट का, जोन ऑफ इक्विवेलेंस, सेम वैलेंसी. तो एंटीजन और एंटीबॉडी बाइंड करते हैं, और जब उनका ऑप्टिमल कंसंट्रेशन होता है जिसको हम जोन ऑफ इक्विवेलेंस कहते हैं, उस समय क्या होता है कि एक मैश लाइक स्ट्रक्चर बन जाता है, एक जाल जैसा स्ट्रक्चर बन जाता है, जिसको हम कहते हैं लैटिस फॉर्मेशन. एंड दिस लैटिस फॉर्मेशन एक्चुअली अपीयर्स और विजिबल टू यू. इसीलिए इसको हमने डायरेक्ट डिमॉन्स्ट्रेशन एंड ऑब्जर्वेशन ऑफ रिएक्शन कहा, जहां पर आप एंटीजन एंटीबॉडी को मिक्स कर रहे हैं, इंक्यूबेट कर रहे हैं, or within no time you can see the reactions. Likewise there are so many different tests, जैसे कि flocculation, एक टेस्ट और होता है flocculation, जहाँ पर insoluble particulate यूज कर रहे हैं एंटीजन्स के लिए होता है. एंड ट्रिपोनीमा पैलिडम बैक्टीरिया एक है, हेलिकल बैक्टीरिया, उसके लिए वो यूज किया जाता है. So if you have any query you can ask me up to this point. If you have any query. Any doubt?
sir.
No, sir.
Okay, so I am now trying to cover one more topic. uh, हम लोगों ने एम्ब्रियोनेटेड एग तो पढ़ लिया था ना, कल्टीवेशन ऑफ वायरसेस इन एन एम्ब्रियोनेटेड एग.
Yes, sir.
Yes, sir.
Yes.
Yes, sir.
Yes, sir.
Okay. Now cell culture. Okay, so we have already discussed the cultivation of virus in an embryonic egg. हमने पढ़ा था एक्चुअली इसको assay of viruses में. तो isolation cultivation of viruses by different method. अब अगर आप इस टॉपिक को देखेंगे तो आप देखेंगे जनरल मेथड्स ऑफ डायग्नोसिस सीरोसिस, जिसमें कल्टीवेशन ऑफ वायरसेस इन एन एम्ब्रियोनिक एग, एक्सपेरिमेंटल एनिमल्स एंड सेल कल्चर. यानी ये बोल रहा है कि वायरस को हम कल्टीवेट कर सकते हैं तीन मेथड से. एक मेथड है एम्ब्रियोनेटेड एग, फर्टाइल एग, जो हमने डिस्कस किया था. We can also cultivate viruses in an experimental animals or animal model or by cell culture technique. Remember, it has a wide application, means that can be used for cultivation as well as for diagnosis and other applications. So this we have discussed cultivation of viruses in an embryonic egg. ये हमने discuss कर लिया था. Now let me take the second point. Yeah, cell culture technique. Yes, yes, cell culture technique. Right. So, in cell culture technique, what we do, we basically what we are doing, we are trying to cultivate the cells in the laboratory. We call it as cell lines, because we know that virus will grow in cells. Animal virus will grow in animal cells, right? And you can only see the growth of viruses by observing cytopathic effect. So for this purpose, what we do actually, the concept is we have organ, right, of a specific origin, जैसे African monkey kidney you have used, by different methods like mechanical methods, chemical methods, and enzymatic method, right? We basically break down these organs into the cells, and we make it the cells as a cell monolayer conventionally. But now we generally procure these cell lines from National Center for Cell Sciences, National Center of Cell Sciences, Pune. यहाँ NCCS पुणे से procure करते हैं. You can visit this website NCCS Pune and you can get the suitable cell lines as per your requirement. So you will get the cell lines, right, in a flask containing suitable medium. So what we require is you need a cell culture flask. For your information, Greener, Tarson, there are so many companies that make a cell culture flask. These cell culture flask varies on the basis of their volume, right, and it is designated as say 75 centimeter square, right? So 25 centimeter square. In case of Petri plates, we differentiate Petri plates on the basis of diameter. Means say volume. What I'm talking about volume. तो आप diameter के बारे में बात करते हैं. इसी तरह cell culture flask जो है, we we differentiate the cell culture flask on the basis of volume. So we have a cell culture flask. It is generally plastic, and we use special medium जिसको हम कहते हैं growth medium. In case of bacteriology, we use the word culture medium. Here we use the word growth medium. There are different type of growth medium which are available. It is generally used ready-made. It is liquid. It contains suitable antibiotics to inhibit the growth of contaminants, especially mycoplasma. Then it contains growth factors like serum. It is sterile. right? Generally, we prefer filtration method for sterilization because it contains antibiotics. It also contains indicators which shows the change of pH. Change of pH. This is why this is important, because you can, if you know the cell lines will grow at particular pH, and if the pH of the medium changes, it may cause the inhibition of the cells. So these are ready-made medium. The examples are Delbaco's modified Eagle medium. There are so many medium readily available. I will show you the picture of this medium. Let me check if I can show you the. Yeah, so you can see this picture, right? So this is, so different companies prepare this, this your high media. There's so many companies. You can see this picture. ये DMEM है, Delbaco's modified Eagle medium, because it's a culture medium. This color is because of the indicator, and change of color confirms the change of pH. So this is ready-made basically. Na? So we use these ready-made medium. So this flask is filled with the appropriate volume of medium and then seeded with the cells. जो cells आपने procure की थी. You incubate it at 37 degrees centigrade at 5% CO2 tension in an incubator. We call it as a CO2 incubator. We have also this incubator, CO2 incubator, right? In case of bacteriology, we use simple incubator. In case this, we use CO2 incubator because carbon is required. And after we can say 12 hours, say 24 hours, you identify the growth of cells under inverted microscope. So under inverted microscope, you will see the growth of the cells. It will spread. It shows adhesion onto the surface. Last के जो surface है, इस पे ये bind हो जाएंगे, and because of contact inhibition, it form a single layer. We call it as a cell monolayer. The growth is called as confluent growth. So you can see the cells of the particular cell lines which is showing. अब आप ये माइक्रोस्कोप में देख रहे हैं, you can see these cells, right? So आप जो माइक्रोस्कोप की स्टेज है, उस स्टेज पे इस फ्लास्क को मूव करते रहेंगे, एंड यू कैन सी द फ्लास्क, राइट? पूरी एरिया को देखेंगे, जैसे ये पूरा एरिया को आप देखेंगे और देखेंगे सेल्स ग्रो कर गई है. So this is how you basically cultivate the cells in the laboratory. Once the cells has been established, it has been grown as a monolayer. Now it is ready for the growth of viruses. Then the next step is you will infect it with a suitable virus, suitable volume, suitable dose. Tissue culture, infectious dose 50 is determined. TCID 50, है ना? So virologist determine the viral dose, and appropriate volume of the viral dose is infected and is then allowed to grow at the same conditions, that is 37, 5% CO2 tension in a CO2 incubator, and post inoculation you observe the growth of viruses. We call it as a post inoculation to 12 hours, post inoculation 48 hours, post inoculation 72 hours. So continuously you will observe the growth of the cells, this growth of viruses, by observing the cytopathic effect. जो मैंने हम लोग डिस्कस किया था, cytopathological effect or cytopathic effect, and the change in the cell morphology confirms the growth of the virus. Once the viruses grow, you can go for further culturing. जिसको कहा था passage, or you can store it at low temperature, that is deep freezing. So suitable cell lines are used. So let me show you the site again. This is for a microbacteriological cultures, we go for this MTCC site, Chandigarh में जो है Imtech, or for this National Center for Cell Sciences, right? So you can contact for any cell line. So about this is given. Yeah, so you will click the repositories. Yeah, so you will find cell repository. Here you will find the cell lines. Once you click it, you will find the repository details where you will find different cells. So how to obtain cell lines? So a requisition form hai. You have to fill it and you have to submit it with a suitable amount of the fees, right? Around 500 something. And then they also show the different service as well. So you can, yeah, cell line deposition in National Cell Repository Pune, ये service में available है. There are different services which are available, and this is showing how this you will do the requisition of the cell lines to the curator. Job processing, cell ready for dispatch. Then it is by speed post or courier, and user will get it, right, and will maintain it. So you can get this from the NCCS Pune. For this is for your information. And the last one is cultivation of viruses in an animal model, right? So generally this whole experimentation that we do in animal comes under a committee controlled CPCSEA. So this is for your information that you cannot handle any animal, you cannot do any kind of experiment in animals without permission. So for this, you need a permission from the committee, and we call it as an animal approved lab. So जब हम कहते हैं animal approved lab means the lab has been taken approval from this committee CPCSEA. Let me show this. So this is called as committee for the purpose of control and supervision of experiments on animals. So before starting any kind of experiment, you have to take permission from this. So they have a complete processing. There is a committee. They review your facility and then give you the permission. So why I'm saying this, because you cannot do any kind of virological experiment in any place without proper consent or permission. So we use generally small animals like Swiss albino mice, white mice, guinea pigs, rabbits, pups, right? The age of animal is important. It should be young. The health status is just first checked before doing kind of an experiment. Then viral strain, passage history, right? Genetic identification of viruses are very important. Again, genetic identification of animal is required. Then route of inoculation is again important in animals. So in the case of virus inoculation in animals, we first identify the site of inoculation. There are different routes of inoculation, like one is intraperitoneal, which is a peritoneal cavity. It is generally this place is preferred when we try to induce the immune response because primary lymphoid organs are there. So intraperitoneal, intravenous. Generally in rabbit it is through marginal ear, and in mice it is through tail veins. Then subcutaneous. In subcutaneous, the thumb and forefinger are used to lift the skin, and the syringe is put in horizontal, and the virus is inoculated by injecting 1/8 or 1/4 inch, and there is a raised area formation at the site of inoculation. इसको कहते हैं bleb, BLEB, bleb appearance. It confirms that the virus has been injected. Then intramuscular, right? and then intracranial, etc. So there are different routes of inoculation. We use specific syringes like tuberculin syringes of 26 gauze. Here this gauze means the diameter of the needle, and the length of the needle is 5.5 by 8-inch needle. So specific syringes are used for the inoculation of virus in an animal model. And we also need expertise. Generally vegetarian are allowed to handle the mice or animals. So these are some ये plastic boxes होते हैं of specific volume, और इसमें specific size का जो box है उसमें number of mice determined होते हैं. This you follow as per the guidelines, right? So simply what you do, you basically have a boxes, having a different mice, 6 mice in each box, and we have a control groups as well. And generally we do this experiment by inoculating the virus at a suitable site. Then handling of mice is again very important. Generally the mice is taken out of this box through this tail. अब आपका जो माइस है उसका जो tail end है, ये tail है, ये tail end है, ये माइस का origin point है, right? And this mice is there. So this is first picked. ये इसको पकड़ते हैं, and the last के 2 fingers है उससे then इस end को पकड़ते हैं, and then जो माइस का जो ईयर पार्ट है, ये दोनों पार्ट है, इसको थंब एंड 4 फिंगर से पकड़ते हैं, राइट? तो यानी कि आप सबसे पहले आपने इससे उठाया माइस को, आउट ऑफ द केज से बाहर निकाला, फिर आपने लास्ट के 2 जो फिंगर्स हैं उस 2 फिंगर से यहाँ पे पकड़ा, और थंब और 4 फिंगर, थंब और फर्स्ट फिंगर से आपने इसको ग्रैब किया. आपको ये ध्यान रखना है कि नेक ग्रैब ना हो, और आप इसको इस तरह से होल्ड करेंगे कि वो बाइट ना कर पाए. बट precaution is important while handling the mice is once you take the mice out of the box, it will try to bite you. So what you will do, ये प्लास्टिक की बॉक्स है जिसमें आप देखेंगे ऊपर स्टील के इस तरीके से रॉड्स होते हैं, राइट? यहाँ पर एक ओपनिंग होती है, एंड दिस ओपनिंग इज टू फीड द माइस. सो ये फीडिंग वाटर लगा होता है, फीडिंग वाटर में भी फिल द सुटेबल फीडिंग मटेरियल लिक्विड्स जैसे मिल्क है ना, जिससे वो सकिंग कर सकता है. तो जैसे आप माइस को बाहर निकालेंगे, निकालने के बाद आप इस जो आपका ये रॉड्स है इसमें बैकवर्ड डायरेक्शन में मूव करेंगे, ठीक है? ये इस तरीके से मैं किसी ने वीडियो दिखाऊंगा, ये बैकवर्ड डायरेक्शन मूव करेंगे, एंड द माइस विल ट्राई टू मूव फॉरवर्ड. सो द माइस विल ट्राई टू मूव फॉरवर्ड एंड यू विल मूव इट बैकवर्ड, देन देयर विल बी स्टैटिक कंडीशन, एंड दैट टाइम यू विल catch the mice. Otherwise it will, if you put it and stationary condition में रख देंगे, then it can move back and bite you. तो इस तरीके से हम माइस को पकड़ लेते हैं, एंड देन एट सुटेबल, जैसे थाई पे आपको इंजेक्ट करना, आप थाई में सुटेबल वॉल्यूम ऑफ वायरस को इंजेक्ट कर देते हैं. सो इंजेक्ट करने के बाद देन यू ऑब्जर्व द माइस, राइट? एंड अगर डेथ आफ्टर इनॉकुलेशन के कुछ पर्टिकुलर टाइम पे होती है तो उसको काउंट नहीं किया जाता है. दैट इज नॉट बिकॉज ऑफ दिस इंजेक्शन, राइट? सो उसके स्पेसिफिक टाइम के बाद, डिपेंडिंग अपॉन द पैथोजेनिसिटी ऑफ वायरस, यू ऑब्जर्व फॉर द डिफरेंट पैरामीटर्स. चेकलिस्ट होगा, जैसे कि वेदर इट इज एबल टू फीड, वेदर इट इज एबल टू वॉक, राइट? वेदर इट इज शोइंग एनी काइंड ऑफ पैलेटिक सिम्टम्स. So if it is showing if it is not feeding or unable to walk, it's showing paralytic symptom, or there is change in the eye movement, right? It confirms that okay, the virus is multiplying, and once it is established that okay, or ultimately death of the mice, so you have to sacrifice it and then to get the specific organ out of that mice, right? And then it is further then subcultured in a cell lines. अब इसके बाद आप उसको cell lines पे grow कर लेते हैं. So the summation is कि you can cultivate the viruses in an embryonic egg, in a suitable animal model, or in a cell culture system. So different viruses are cultured by different methods. Different modes of inoculation are used, different protocols are used, and the ultimate idea is virus once multiply in the system, living system, it shows effect, and then you can either store it or you can transfer it or passage it or yeah, subculture it. So this is how you can confirm the growth of the virus. So any doubt, anything you want to ask, you can ask me.